Okay, so welcome to chapter 1 in human biology. So we're going to be taking a look at some of the preliminary ideas in biology. So first of all, when you take a look at biology, the important thing to understand is that in the study of biology, we are studying life. Living systems basically is what we're studying. And so when we take a look at living systems and the environments that they're in, first of all, there's some hallmarks of living systems. We all know that they have organization to them. So they're ordered systems. They need materials and energy. They're homeostatic, which just basically this means the term homeostasis. We're going to see this again in the future chapter in chapter 4. This basically means the maintaining of constant internal conditions. So you kind of have a constant stability internally. That's homeostasis. That allows us to respond to stimuli. So we're adaptive, we're responsive. And then ultimately, we reproduce and we grow so that we can ultimately reproduce. And then over generations, of course, then we have a history of change, and that's evolution. And the important thing to remember is that all living organisms have to do this. They all have to basically do all of these, and the holy grail of all of this is reproduction. So this is actually the reason, the purpose of all living organisms on the planet is to reproduce. And everything that happens in the body is for the goal of reproduction. It's so that you can build a reproductively competent body. And so everything in the body pulls toward that. And so these are what I call, this whole list taken together is what I typically call the big biological questions of life. And so since they're the big biological questions of life, what that basically means is that you have to answer all these. You have to answer these. And that's what biological organisms are doing. So whenever they're doing some sort of a biological mechanism or a process, it's because they're trying to answer one of these big biological questions of life. And the reason why they're trying to answer those big biological questions of life is ultimately because they want to reproduce. And that's the goal of a biological organism. So before we begin into the deep woods of biology, let's talk a little bit about where it all starts, because biological systems are built on principles, they're built on foundation. And the foundation comes in two different forms. You have a foundation of physics and you have a foundation of chemistry. And so when you take a look at biology, there's what we refer to as levels of organization. So basically, when we take a look at everything, ultimately everything is made out of smaller parts. And ultimately, we are all made out of atoms. And so typically what happens is atoms, which are essentially the elements of the universe, oxygen, nitrogen, things like that, that essentially pepper themselves throughout nature. These atoms come together and they join to form molecules. Here's a good example. So let's take an atom of hydrogen, which is a pure element in the universe, and some oxygen. Now, when you put these two guys together in the form of a chemical bond, which is that little line that I just drew there, then you're starting to form some associations, some relationships, if you will, between these two atoms. And so oxygen, as it turns out, can actually form two of these relationships with hydrogen. And so this gives us now a molecule. So each of these individuals is the atom, and then, of course, the entire thing is a molecule. And in this particular case, the molecule happens to be H2O, which is water. Now, these molecules, that's a pretty small molecule, but you can get larger and larger molecules, what we refer to in biology as macromolecules. This is typically the things that biochemists will study. And as these macromolecules come together, they start to form cells, which is the smallest functional unit of life. So this is, we oftentimes refer to the cell as the basic unit of life. Now, what does that mean? Well, it means basically this. So what this means is that this is the smallest that we can get. And still be able to answer all those big biological questions of life that we just talked about. And so that's basically cells. So some organisms are single cell, like bacteria, for instance. Other cells are multicellular, like us, humans, and dogs, and things like that. They have many, many cells. And when you have many, many cells, oftentimes you have to organize yourself. Remember, organization, that's basically coming back at number one on our list. Once you have multiple cells, you have to organize them, and we organize them into tissues. So basically, tissues are groups of similar cells that perform a particular function. Okay, so they're similar. They're kind of like on a particular team, if you will. And so this is kind of what this looks like. So if you take a look at this from sort of the bottom up, and I'll go ahead and slideshow this one so that you can actually see it a little bit better. So here's your atom level, which is the smallest unit of an element. It's the smallest that we can get. And then atoms will come together to form molecules. In this case, we have a molecule of methane. Those molecules will come together to form cells. So here's a good nerve cell, a plant cell. Those cells will organize themselves together to form tissue. So here you can see multiple nerve cells together to form nervous tissue, multiple plant cells together. They're associating with each other to form plant tissue. And then having multiple tissues come together to form an organ. In this case, the nervous system is forming the brain, and the plant tissue is forming what could be becoming a leaf. And multiple organs together, cooperating together, will create the nervous system. For instance, the brain is only one organ in the nervous system. You'll have the nerves themselves, you have the spinal cord, and various other sorts of things that form the overall nervous system. In the plant, the organ is the actual shoot system. So those branches with lots of leaves on them. As you put together all these organ systems, we get the organism. This is us. So in an individual human being, we are a collection of organ systems. So we've got a system, an endocrine system, got a muscular system, a skeletal system, and so forth. And all those organ systems together basically will form an individual. And of course, all these other systems together, like your root system, your shoot system, and so forth, will form the ultimate tree. Now, once we kind of move forward, we define organisms or groups of organisms in terms of species, which is a group of similar types of individuals. So for instance, we have one species here, that's us, that's Homo sapiens. And then we have another species here, which is a species of tree. There's another species here, which is the grass, and so forth. But each species is a group of interbreeding individuals. That forms then and kind of leads to a population. So oftentimes population and species are fairly similar. And so a population will basically be larger groups of the same species. So species and population are very similar to each other. They're very synonymous to each other. Just a population is a matter of scale. So it's basically a larger scale. So you have more people as opposed to just a few. And then if you want to add more to it, you move on to community. And in community, basically you have a bunch of interacting populations of different species. For instance, here you have one population of Homo sapiens, one population of zebra, one population of trees, one population of grasses. And if they're all interacting with each other, then that's basically forming a community. So the zebra will eat the grass, that's an interaction. Maybe they'll strip some of the leaves off of the tree, that's an interaction. The humans may hunt the zebra for meat, and that's an interaction. So communities are interacting populations, oftentimes with herbivory and carnivory. So those are all community interactions. Now, if you kind of scale up community to the ecosystem level, notice we're getting broader and broader and broader. Now what you've done is you've basically added your community interactions, and now you've added a physical environment. So the ecosystem basically has both living and non-living environments. So for instance, you can see there's a lot more people here, but now you can see the mountains. So for instance, terrain, so rainfall, soil, things of weather, those are all what's called abiotic non-living. So when you start to have this issue of organisms interacting with their non-living environment, then you're talking about an ecosystem. And then multiple ecosystems will basically lead into our biosphere, which essentially is the Earth. Okay. So that's kind of a pictorial of our levels. So we left off at tissues. So tissues are organized into organs, and you have several different types of organs. A good example I like to use here is the liver. So the liver has multiple tissues. It has both what's called hepatic tissue, that's the actual liver cells. It also has vascular tissue. There's blood vessels going through the liver. It also has nervous tissue, and it has connective tissue. So there's lots of different tissues in the liver to make up the liver. And then the organ system is basically a collection of organs working together. For instance, the liver is part of the digestive system, which also houses the stomach and the intestines and so forth. Now, along with the digestive system, the nervous system, the endocrine system, and all the rest of them, those organ systems will come together to form the organism, which will be the individual. And then those individuals interbreeding with each other will create the species and the population level. And then those populations interacting with each other will create community. And then from community, as they interact with their non-living environment, that becomes the ecosystem. So when you're talking about the environment, how plants, say, for instance, get soil nutrients, or how they behave in rainfall or drought, then you're talking about an ecosystem level interaction. That's non-living stuff. How do we deal with non-living stuff and non-living aspects of our environment? Then, of course, all of these together will be the biosphere of the Earth. Now, one of the important aspects and the themes of life is energy, right? So energy basically is something that we need in order to be able to do anything, really. Anything biologically, we need energy to do it. So energy, by basic definition, is the capacity to do work. And so this allows us to be able to do stuff. So when we eat, we have to acquire materials to build our own body. And energy in the molecules. So we get materials and energy, and some of those materials are nutrients. These are things needed for energy and building blocks, but they're also needed for the chemical reactions in our body. So you have to eat so that you can acquire the necessary components that you need in order to get your body to function properly. Now, what about these chemical reactions I just mentioned in your body? Well, basically, your body is a gigantic reservoir of chemical reactions. And all those chemical reactions in your body we refer to as metabolism. So it is essentially the sum of all the chemical reactions that are inside your body. Now, if you're a single cell, it's inside your cell or the organism. And so typically, most of these need energy in order to do these chemical reactions. So hence the reason why energy is an important concept for living organisms. Where do we get our energy from? It's very simple. All the energy in life on the earth comes from the sun. That's as simple as that. That's its origin. But the problem is the energy is not accessible to us, especially as animals. So we need a strategy to be able to acquire or to capture that energy in a usable form. And that's where we get to the all-important process called photosynthesis. This is basically what's happening in plants primarily, algae, of course, and some bacteria. But basically what this is going to do, this basically captures sunlight energy and converts it into chemical energy. So it harvests energy from the sun and converts it to chemical energy, which is a type of energy that we can use. And that's because we are chemical systems. So what do you get out of photosynthesis? You basically produce sugars, so glucose, for instance, which is essentially the primary food source, the energy molecule for all other organisms. So really, it's photosynthesis that kind of makes it all possible for all the rest of us. Okay, so the next topic, if you're wondering how this is flowing, I just want to draw your attention back to our list, right? So we've talked about organization. We just talked about acquiring materials and energy. Now we're going to be talking about homeostasis. So we're working our way through our 6-part list. So if you're wondering how this is working out, that's what we're doing. So homeostasis is important for living organisms because they have to basically maintain a constant internal environment. And this allows them to be able to do chemistry. In order to do chemistry, you have to know what you have. You have to add, say, for instance, equal parts of A to B. And so you can't have a lot of variables in there. It has to be maintained and constant. And so most of our systems are looking toward and moving toward maintaining even homeostasis. For instance, our temperature, core body temperature, is homeostatically set at 37 degrees C. And so what we try to do is we try to maintain our body temperature in that. So for instance, if we start to get a little too hot, then we start to move toward sweating to cool us off. If we start to get too cold, then our muscles will shiver to produce heat. So what you're trying to do is you're trying to maintain that core. You get too cold, you've got things that you can do to get back. You get too hot, you have things you can do to get back. And that's kind of what it's like. So typically a homeostatic situation should look a little bit like a sinusoid, like an ebb and flow, right? So for instance, if you get a little too high, mechanisms will turn on to bring you back down. If you get a little too low, mechanisms will turn on to bring you back up. So you're kind of constantly going back and forth, oscillating like the tide, coming in and going out, coming in and going out across what is basically a homeostatic norm. And it's a range, just like the tide. So the next on our list is response to stimuli. And this is important because we wouldn't be able to maintain internal conditions unless we responded to stimulus, right? Because the idea is if your body's getting too cold, you have to have some sort of a way to sense that, right? In this case, we're going to use the hot stove. So if you have your hand on a hot stove, it's basically too hot, obviously. But one of the things you need there is you need some sort of sensory information to tell you that your hand is burning. And then the next thing that you need after that is some sort of an integrated, reasoned out response. You have to have some sort of a plan for that. And so the idea is your nervous system is doing this all the time. It's taking this information in, it says, you know what, it's too hot. And then your response is pull the hand away, right? So that's external. That's our response to external stimuli when things happen to us. But we also have internal stimuli that are happening. For instance, these are basically internal sensors that we're not even aware of half the time. And these guys will relay info to maintain homeostasis. So for instance, adjustments to your blood pressure. So your blood pressure naturally goes up and down all day long. And so as it gets a little too high, you have internal sensors, which will basically relay that information to your brain, and then it'll adjust that blood pressure back into these homeostatic normal ranges. So you have any number of homeostatic things. So a lot of things. Pretty much, if you can measure something in your blood, whether it's blood pressure or calcium or anything like that, if you can do a blood test and measure it, it's under homeostatic control. And there are stimuli, there are responses and sensors to those adjustments. So as those fluctuate in your body, there's sensors that sense that fluctuation, and then they relay that information to elicit a correct response. Okay, so the next one on our list is going to be our reproduction and development. So basically, we reproduce, we're going to be passing our genes on to the next generation. So we have half of our DNA comes from mom and the other half comes from dad. And then when we're born, the first thing we have to do is we have to grow. So to grow, we have to increase in size and in the number of cells. So typically we start off as one cell, and then we have to turn into a multicellular individual. So a lot of what we're doing early on is we're growing, we're just increasing in size and getting bigger and bigger. Now, as we go through different stages in life, our body needs to do different things, and that's development. So ultimately, there's different types of developmental periods. We tend to think of development as embryonic, but we also have fetal development. We have newborn development. You growing through development as you're a kid. Your brain is developing as a middle schooler and as a teenager. When you're a young adult, you're developing as a young adult. As you start to move into middle age, you move into a different stage of development in your life as you move past childbearing years. And as you move into older age, you move into yet another developmental period. And ultimately, it basically, we kind of go in and out of these different developmental stages throughout our life. And so what we refer to these as like our childhood, our adolescence, our adulthood, and things like that. And so these are our major developmental shifts that we have and that we go through. And it's not just psychological development. Oftentimes people will think of, childhood and adolescence, things like that, psychological, but this is also biological. So your body is actually transforming from a child's body to an adolescent's body, from an adolescent's body to an adult's body, and so forth. So these are actually biological as well. The other thing that we also need to mention when we're talking about reproduction and development is the actual genetics, the information that basically makes us all work, and that's our DNA. So our DNA is basically the genetic information that makes us all work. This is essentially all the information needed to build us from scratch. So every scrap of information, every even little jot and tittle, even just every little dot and cross T, everything, every little itty bitty piece of information that's necessary to build you from zero to where you are now is encoded in your DNA. It is the ultimate master source of information. And so it is the master source of information that directs all of the activities of your body. It doesn't just get passed on from one generation to the next during reproduction, but it actually also orchestrates how all of your cells function and how they're actually working. So it actually is the main organizer, if you will, the command center of all of biological existence. And your DNA is broken up into short pieces of information called genes, which are short segments that specify a particular trait. So for instance, you have eye genes, skin color genes, perhaps hair color genes, things like that. So these are specific sequences of DNA that are specially encoded to govern and to control a particular trait. So genes control traits. Now, if you were to alter the sequence of these genes, then you create what's called a mutation. So a mutation is basically the altered sequence of a gene. Now, generally speaking, mutations are mostly negative. They can be beneficial, and when they are, they can give an organism something to improve itself with. But the problem is, it's a big but. Mutations are mostly very negative. So there's not very many beneficial mutations that we've documented. Most of them are very negative. We have tons of information on negative mutations, but not much for beneficial mutations. Now, of course, the last piece then on our list is our evolutionary history. And so there's a couple of different things that I just want to talk about a little bit. So evolution essentially is very simple. It's how a population changes over time. And this is typically a genetic thing, right? Because the way an organism changes, the information that it encodes its body plan is encoded in its DNA. So for an organism to change over time requires its DNA to change over time. So evolution is fundamentally a genetic thing at its heart. The idea being that there's several different mechanisms of evolution. That is to say, how do organisms change over time? Well, one of those mechanisms of evolution was mutation. So that was one of the big mechanisms of evolution. And as we've sort of gotten a little more sophisticated in our thinking, we've kind of expanded a little bit. Another big one is natural selection. So that's another mechanism of evolution. Now, here's how this works. When you get a mutation, it's either going to be good or bad. So if it's good, then it will favor you. And the idea is that you'll survive better and that you'll reproduce more and that you'll pass those genes on. It's reproductive success. If it's bad, then you'll be basically wiped out of the gene pool. Think about it this way. When a new variation occurs, let's say, for instance, we get pink zebras. That could be either a good thing or a bad thing. So if you're a pink zebra, one could automatically think, well, wait a minute, that's like a negative thing, right? Because now all of a sudden the lion sees this bright pink zebra and it makes you stick out. So guess who's at the top of the lunch menu, right? So that's you, pink zebra. So in that case, it's negative if it leads to greater predation. However, what happens if in the zebra world, you're looking pretty good? Pink is pretty amazing. It's like, wow, look at you. You are a one good looking zebra, you pink zebra. And everybody wants to mate with you, right? So if you're kind of the hot commodity, then this could also work in your favor. So this would be positive selection. Because now you're going to have more little zebra babies. You're going to pass pinkness further throughout the zebra. And then eventually over time, you'll start to see pink zebra herds. And so it depends on how that looks and how that works. But basically, when you take a look at genetic variation, natural selection simply just sifts through and sorts through genetic variation. Now, generally speaking, what that means is, remember I mentioned that mutations are by far more negative than beneficial, which means that most of the time, if you get a mutation, you're getting rubbed out. And so natural selection will essentially eliminate you from the gene pool. And so that's typically the case. There's also adaptation. In terms of adaptation, this is a situation where you adapt to your environment. But oftentimes this is better understood with like if we have pre-existing, and what I mean by that is let's just kind of, rather than going all the way back and saying that all genetic variation arose from mutation, which is what an older version of evolution says, is that everything, all changes are the result of mutation. Let's imagine that we have a pool of variation. Let's go back to the family, like the dog family. We're talking about all coyotes, dingoes, hyenas, jackals, foxes, wolves, common house dogs. So all the dog things, the canids at the family level of the canids. So there's already like a big pot of genetic variation at that family level. So basically, let's take a look at the family Canidae. This is the dogs, right? So there's already a lot of dogness in there. What adaptive radiation does basically is it says that if you have this sort of common dog, then each of these there's a lot of genetic variation that you can already adapt to. So for instance, you don't need to create a bunch of new mutations because there's already a lot in there. So for instance, in some environments, some genes may be more favored and may be selected for. So for instance, in some environments, you may end up getting things like the wolf. In other environments, you may be getting, say, for instance, the coyote. And still yet other environments, you may be getting, say, the fox. And then, of course, our common beloved pet, the house dog. But basically, they're all essentially radiated. They're all sort of adapted from this one big pot of dogness, of dog genetics. And that's kind of what adaptive radiation is. The idea, if you kind of have this big pool of genetic variation, that you can just adaptively radiate from that. And the big story behind that one that we understand from that one is, if you just take a look at the house dog, look what we've done to the house dog, just a basic pet, Canis familiaris, right? If you take a look at that, I mean, we've gone from a single dog, which is what happened about 3,000-4,000 years ago, right? There wasn't a whole lot of house dogs, and we've turned this thing into like a mastiff, and we've turned it into a teacup poodle and everything in between. So what that basically tells us is there was a ton of genetic diversity in that one single dog that was capable of turning that thing into some drastically different types of dog breeds. So that's a lot of genetic variation. It just adapted, and we just sort of pulled out that information. That's what adaptation is. Only in this case, adaptation is the environment pulling out that genetic variation as it's adapting to its environment. Another good example of that would be the bears, the great bears, right? So you have polar bear, grizzly bear, and black bear. So there's a good example of 3 different bear traits that were adapted to their environments. And so that's kind of a thing. And so we've already talked a little bit about this, right? Because when you start taking a look then at, of course, evolution of organisms and the relationships between each other, then it's not too long before we have to start talking about classification, right? So how do you classify these organisms? You got a ton of them out there. And so how do you classify them? And so what we have is this whole division of science called taxonomy, which is essentially the classification of organisms by morphology, or their body, what their body is like. Okay. It's like a 20 questions of biology, right? So you're just asking questions. It's like, are you this or are you that? Are you this or are you that? You did basically asking a bunch of questions. The first question that you ask at the very top is ultimately, are you a prokaryote or a eukaryote? That's the first question you're asking. Now, to explain what those are, is every single cell on the face of the planet is broken down into one of 2 types. You're either a prokaryote, in which case typically you are going to be single celled and you're lacking a nucleus. So that's your prokaryote. Or you're going to be a eukaryote, in which case you have a nucleus and you've got organelles, and oftentimes you're multicellular. So that's the difference, right? Whether you have a nucleus or not, or whether you have organelles or not. Now, of the prokaryotic ones, we basically broke ourselves into 3 major divisions: 2 prokaryotic and 1 eukaryotic. So the 2 prokaryotic domains were bacteria, archaea, and then eukarya was the eukaryotic domain. That is to say, it had a nucleus. Now, the reason why archaea is kind of in the middle is because there are some things in archaea that look very bacterial, like it's a prokaryote, for instance. There are some things in archaea that look very eukaryotic, and in some of the ways that they do some of their processes are very eukaryotic. So they're kind of somewhere in between. So we basically think of the world as split into 3 different domains, and all living systems and living organisms will be one of those 3 domains. So here's kind of how it looks. So basically, when we take a look at taxonomy and evolution, what we essentially have is, you have to remember, typically all of this stuff here, all of this here doesn't exist. I mean, to say it's not alive, right? So what we have is basically a bunch of organisms here, 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 and here. And the idea being is that typically, kind of like our familial sort of thing, right, is the more closely related you are, the more likely you're going to look the same. So if you look similar to somebody else, the idea being is it means you're more closely related to them because you share the same genetics, so you're going to have similar looks. And this is kind of something we see throughout the world, right? So for instance, this is kind of in our ethnic breakdowns, for instance. So we have somebody, who looks German, for instance, because even though 2 Germans look similar, it's because they have kind of a common genetic background coming from that particular area of the world. Or siblings, if you want to continue to get even more closely related, for instance, will oftentimes look like each other because they share the same genetic parentage. The idea being that the similarity or the morphology of how you look and your body plan, your body structure, the more similar it is, then it's likely that you came from a similar source, a similar genetic source. And that's kind of the idea behind this. But there are problems to that, because just because you look the same doesn't necessarily mean you're anywhere closely related. And so there are some huge caveats that we need to become careful of. So in our kingdom, in our eukarya domain, we have then broken down into what's called kingdoms. So our kingdoms are basically 6 gigantic groups. So we have bacteria, kingdom bacteria. So these are the kingdoms. We have kingdom archaea, and then we have kingdom eukarya here, which is broken into domain eukarya, broken into kingdoms of protista, plantae, fungi, and animalia. So you can see we're at our 2nd level now of taxonomy. So we've gone from 3 domains, now broken into 6 large kingdoms. And the eukaryotic ones are plants, fungi, animals, and protists. And protists are basically single-celled eukaryotes. Some are plant-like, and some are animal-like. And then we can break these kingdoms up into further and further pieces. So for instance, when you take a look at your domain archaea or archaea, these guys are prokaryotic, but they can do certain things that are very non-bacterial. Of course, you have your domain bacteria. This is an example of a bacterium. These guys are prokaryotic. They can either do photosynthesis or they can do chemosynthesis. They can break apart chemicals and things like that nature. Then you have your eukarya. You have your main kingdoms of eukarya. These are all the cell nuclei carriers. So here's a kingdom protista. These are the different single-celled organisms that you oftentimes will see in pond scum. So these guys are oftentimes pond slime and scum. So whenever you see like a green scummy pond, there's probably a lot of protists in there. Okay. Then of course, kingdom plantae. This is all plants basically. This is including your flowering plants, your non-flowering plants, right? And then of course, algae. Some types of algae are in here. Some types of algae are actually colonial protists. So they kind of split both ways. Ferns, mosses, things like that. Typically, these are the photosynthesizers. So that's where the plants are. And then we have our kingdom fungi. So this is our mushrooms, yeast, molds, things like that. These guys typically tend to absorb their food. And so, as opposed to eating it or making it for themselves. And then, of course, the one we're probably most familiar with, which is what we're going to be spending most of this semester focusing on is Animalia, right? So this is basically from sponges to worms, insects, fishes, frogs, turtles, birds, mammals. So that's kind of like in order of taxonomic complexity. So we have to ingest our food. So we have to eat our food. Okay. So and so when we take a look at kingdom Animalia, then many of us, not all of us, many of us, and animals are invertebrates. What that means is there's no backbone. Now, that's not true for us, obviously. We have a backbone. But when you take a look at invertebrates, they don't have an internal skeletal support structure. So they don't have an internal skeleton like vertebra, backbone. Typically, this is all of our insects. Our insects are huge groups of animals. There's a lot of us out there. Earthworms are soft-bodied. Mollusks, like basically mussels and clams, right? They have a soft body inside of a shell, but they don't really have a backbone. And then, of course, you have the vertebrates. These basically are the animals that have a backbone. And so typically the reason why we have a backbone is because we're protecting something. Inside that backbone, and for us especially, is our nerve cord, which is part of our nervous system. And that's basically protected by that vertebral column, which is our backbone. So for instance, fish have a backbone, reptiles have a backbone, amphibians, birds, us, right? Now, some of us backbones are going to have hair and fur and mammary glands. And if that's true, then we are the mammals. This is our humans, raccoons, seals. So oftentimes they'll give live birth, but not always. Some fish give live birth. But if you have mammary glands, if you nurse your offspring, then you are a mammal. So you can see how what happened is we basically broken these out, and we're kind of breaking them further and further down into different classes and categories, different piles, right? You have a backbone, you're over here. You have no backbone, then you're over here. If you have a backbone and you're furry and with mammary glands, we're going to put you over here. And if you don't have mammary glands and furry, but you still have a backbone, we're going to put you over here. So we're kind of like organizing them in little piles. And that's kind of what taxonomy is all about. So, and that kind of helped us to organize life in general. So now let's take a look at the scientific process. All right. So when you take a look at science itself, science is essentially something that we use in order to understand the natural world. And that's basically what we're looking at. So it can only speak on natural law. That's all it can do. It's defined that way. A couple of things that are important about science that are really critical is that in science, you have to be objective. You can't be biased. And it can't be subjective. That involves judgment or multiple solutions. So it can involve your judgment, because if you involve judgment, what happens is this leads to bias. And if you're telling me something based on your bias, all you're telling me is an opinion. And probably most of us have interacted with opinions well enough to know that opinions oftentimes are wrong. Just because you agree with an opinion doesn't necessarily make it factual. So objectivity is something that science really, really focuses in on. This is what it really sets it apart. Okay, so it has to be objective. They also have their own process to understand the natural world, because the natural world is complex. There's a lot of moving variables and a lot of moving parts. And so what they have is a scientific method, and we're going to take a look at this here in just a second, which is a methodology to be able to understand and think about complex problems. It's a series of logical steps that leads us to a conclusion. The first one, it all starts with, is observation. So observation basically is a way of looking at the natural world and make some sort of an observation. For instance, we'll typically use our senses. So we see something, we smell something, We can use instruments like microscopes. We use a microscope or a tool in order to see something clearer. And then oftentimes we observe this through research. Now, once we've made this observation, then we want to lead ourselves to a hypothesis. But before we get to the hypothesis, what leads us to the hypothesis is step number 2, which is a question. Can we ask some sort of a question? Like, for instance, what makes the leaf green, for instance, if you observe the green leaf? Right? And the hypothesis is a possible explanation for that observation, for that question, right? So it's not correct. It doesn't have to be correct, only testable. So it's okay if your hypothesis is wrong, because the idea is you're just trying to learn. It's your best guess, right? This is your best guess. Your best educated answer. To your question. So for instance, if our leaf is green, let's imagine that we have somebody like way back in the day who's just an artist and they don't know anything about science. So they don't know anything about chlorophyll or anything like that. So they say, well, my best guess is that the leaf is painted green. That's all I know. I'm an artist. That's all I know, right? So for them, their hypothesis, which is going to be number 3, would be the leaf is painted. Obviously, that's wrong, but he didn't know that. But it is testable, right? Now, then once you get past that stage, you're ready to test it if it's testable. Now, important thing to understand about science is we use a couple of different types of reasoning. So inductive reasoning is basically when somebody uses combination of facts into a cohesive whole. Another way to look at this is from multiple observations. You derive a conclusion. An example is you conclude that the sky is blue. Why? Because you've seen the sky many times. So over the millions of observations that you've done of the sky, it's always been blue. And so you can comfortably conclude that the sky is blue. That's inductive reasoning. Now then, from the hypothesis, typically we want to generate a testable prediction based on that hypothesis. These are if-then statements. So let's use our example here. Our hypothesis, the leaf is green. Now here's a prediction, right? So basically, if the leaf is painted, then, notice the if then it should rub off, right? That's a prediction that you would get if it's painted. If it's not painted, then it should not rub off. So here's your if-then statements. And you can test for these, right? Because the next thing you do is, look like a duplication. Okay, hold on one second. Looks like PowerPoint is about ready to crash on me, so just bear with me as it decides what it wants to do, because everything is coming up. Okay. Hold on one second as I reboot PowerPoint. Just bear with me. PowerPoint does this from time to time. It happens all the time, unfortunately. Drives me nuts, but it is what it is, I guess, right? Okay, so here we are. So we just talked a little bit about testing the prediction. So we have those if-then statements, right? And then what we do is we test it. And so we run experiments. So this is, what number am I on now? So the prediction is 4, right? Those are if-then statements. And then we have 5, the testing or the experiment. We design an experiment in order to test the different variables, right? And so oftentimes this is what's called deductive reasoning. It's if-then logic. If this is true, then this should be the case, right? So that's deductive reasoning. And so typically when we organize an experiment, we generate 2 different groups. We create what's called test groups and control groups. And these guys are basically the same, except for the variable being tested. So oftentimes, like for instance, if I were wanting to test a fertilizer's ability to cause wheat to grow, what I would do is I'd create a control group and an experimental group or a test group on the same field, in the same plot, with the same soil, the same rain. And in the control group, I would basically grow my wheat without fertilizer. And in the experimental group or the test group, I would basically grow it with fertilizer. And if all things are the same, then the only variable that could have caused the greater growth would be fertilizer. And that's kind of how you use test groups and control groups. Because otherwise, if I didn't use a control group, I could argue it's like, well, it's not the same. Because maybe it was, the soil. Maybe there was more rainfall. Maybe that's what caused it to grow more. Maybe there was more sun. Maybe the other field was in shade. There's all sorts of things that could have been other than my fertilizer. So if I want to see the effect of one variable, I have to control or keep all the others the same between the 2 groups. Another thing we also use is model organisms, like mice, for instance. And we use these as systems to apply knowledge learned to humans. For instance, drug development is oftentimes started off in model organisms because it's unethical to test things on humans, but we can test them on mice, and then we can transfer that information to humans. So if it works on a mouse, there's a good chance it might work on a human because we're both mammals. As you do your experiment, part of that process is both analyzing and collecting data. So from your experiments, you're going to be generating some sort of data, and you have to present that in different formats. You can use graphs and things like that if you're generating numbers or observations, for instance, like this. Here's an example of somebody who was measuring blood cholesterol levels by week. And so what they saw was that there was a variation in blood cholesterol levels from week to week. And so that's the data that they collected. And then they were able to present that across their graph. In order to describe our data, we use statistics. That's a way we use to describe and interpret our data. And typically when we have statistical data, that basically means that there's a lot of numbers, but there's going to be a source of error. And so we have to be honest about describing that to see just how significant is that number. We do this all the time, right? I mean, especially in an election year when we have all these polls and it's like, the error is like plus or minus, 2% in a poll, right? So for somebody who's like running, neck and neck, like they're both at roughly, 49, 40, 50, whatever percent, and the statistical error rate is plus or minus 2%, then statistically they're kind of pretty much neck and neck. And that's because you have that amount of uncertainty when you're talking about data and the trends in data. typically speaking, if we want to know whether or not a number is significant, that means if it means something, or if it's actually just random scatter, we have to start using things like probability and things like that in order to prove that we have statistical relevance, that this is a relevant number. Like, we didn't just do this experiment and we got nothing. This is actually relevant. This is relevant to our conclusion. And so we have a lot of that in science, and it's important for that. And of course, if it all looks good, then we write it all up and we put it and send it to a publication so that we can get credit for that and let everybody else know what we found. And then from that, of course, from our data, we're going to be generating a conclusion about whether or not our hypothesis is not, it's not correct or not. It's whether or not it's supported or it's consistent with our hypothesis. Okay. And then oftentimes what will happen is that'll lead us to another hypothesis, and then we just kind of keep on going. So this is basically how science works. This is the process of how science works. And every time we learn a little bit of information, we add it to our hypothesis, and eventually our hypothesis grows up into a theory. And so a theory basically are generally accepted explanations for how the world works, right? So for instance, often having been supported by lots of experimental data. So that's what happens. So a hypothesis that is constantly supported or validated through experimentation eventually becomes a theory. And then, like cell theory, for instance, and so forth, atomic theory, things of that nature. And then a law or a principle is basically so accepted that it's pretty much 100%. So this is basically 100% certain. Like, for instance, the law of gravity. The one thing I would disagree with Mater here is that evolution is not a law. The only laws that exist are the laws of physics. So nothing in biology. Why? Because the systems are designed to be flexible and adaptive. So you're not going to have a law if you're designed to be flexible and adaptive. So these basically are essentially unbendable. If you want to use kind of a more poetic term, immutable. That means that they are always true. That if you let go of a pen in midair, it will drop to the ground. That's the law of gravity. That if you add 2 + 2, it will equal 4. There is no alternative to that. There is no such there's no such level of that in biology. Biology is designed, even in evolution, biology is designed to be adaptive and flexible. And so the laws are reserved they're pretty much for physics. So I would strongly disagree that evolution is a law. It's nowhere close, not even close. The closest thing to a law in non-physics would probably be atomic theory, the atomic theory. But certainly nothing in biology. So that's a huge overspeak. So basically, when you take a look at a controlled study, this is kind of an example of a controlled study. So you have a hypothesis. You have antibiotic B is better than the currently used antibiotic A. So that's what you're saying. That's what you want to test. So the first thing you want to do is set up your control groups and your test groups, right? So the control groups are going to be taking a look at, say, for instance, ulcer medication, right? In this case. And so the control groups are going to accept a placebo. So that basically, they don't have any medication. So everything else is the same, right? And then, of course, the test group will so there'll be a test group for B and then another test group for A. So you'll have a test group for A and B. And so then we do, in a lot of times, we do this a lot in drug development, called a double-blind study. And the reason is basically this is to remove bias. So you don't know which one you're giving. So what happens is the doctors don't know this, nor do the patients, which group they're in. The doctors are just giving the patients a pill. It's going to be one of three things. It's either going to be A, B, or just a sugar pill or something like that, or just a placebo, no medication. They don't know which one they're giving. So that's a double-blind test. Two people are blind to this. The only people who know what it is are basically the ones who are tabulating the information in the end. And this basically keeps everything unbiased. And the conclusion then from that would say that antibiotic B had better results. Then this suggests, because we did the double-blind study, we removed bias and we did a controlled experiment, it suggests that there is some truth that antibiotic B may be better than antibiotic A. Okay, so that's kind of how we did this. So here's our statement of our hypothesis. We performed the experiment. So here's basically our three different groups, test group 1, 2, and then 3. And then when we collect the data, we can see that test group 2, which was antibiotic B, did better statistically significantly than, because here's your error rate right here. So you can see that it did better than the error rate. So it's not just random error. For those of you who are interested in baseball and do batting averages, you know what I'm talking about, right? An average can sort of float around. It doesn't necessarily be a depictor. It has to be significant. So here you can see that it looks like it is significant. And it's better than A. Okay. Now, what about science and society? And this is going to be the end of the chapter, just a few slides, just to kind of pull it all together. So science and technology, certainly, right? So when science, take a look at science, this is basically our systematic way of looking at learning about the natural world. When we apply that, that's technology, right? So oftentimes when we learn something in science, we apply it to something and we're able to develop technology. For instance, that's how we developed the cell phone. That's how we developed this new drug. When we learn something, then we're able to apply it. And that's the power of science, is that science, unlike any other discipline, has the ability to improve by applying its knowledge directly to technology, which then improves our lives. So if you take a look at science in terms of the kind of the example of climate change, right? So for instance, in climate change, we basically have these sort of normal cycles of climate, typically in the earth. And then of course, we have these changes that are happening because of human activity. And oftentimes it's because of carbon, oftentimes in the form of CO2 increases. And then what this does is, because carbon traps heat, it's a greenhouse gas. Because it traps heat in the atmosphere, what happens is you start to get this global warming, the increasing temperatures because of that. And so as you start to get abnormally warmer and warmer atmosphere, then the CO2 levels are causing global warming, and the global warming is going to start changing the Earth's ecosystems. Why? Because weather patterns are driven by both atmospheric and water temperature changes. So a good example is, how do we know it's going to be an El Nino year? Well, we know that because the surface temperatures of the water just off the Pacific coast are abnormally warm. That basically means that those abnormally warm temperatures in the ocean are going to change the atmospheric climate it's going to start creating different weather patterns and affecting the overall atmospheric conditions above it, both hydrologically, that is to say, the way the water is evaporating, it's going to be evaporating more aggressively, creating more rainfall in some places, and it's going to be shifting the movement of the atmosphere, because one of the things that happens is when you get wind, for instance, you basically have the movement of air from a high position to a low position. For instance, we think of it as a high pressure versus low pressure system. Oftentimes, high pressures are associated with heat and cold. And so as air increases in temperature or decreases in temperature, it tends to move. So for instance, we have all heard this term that hot air rises, cold air sinks. Well, if you use that, and we kind of know this, right? Because in the middle of a hot day like today, if you have a multiple story house, the bottom levels are cool and the upper levels are really hot. As hot air rises, cool air sinks. Now, if you put that on a climate scale, then if you have air moving like cold air that's sinking to the ground, that's going to create wind. And so as air moves from an area of cold or hot, that basically creates wind, but it's moving around and it's taking with it all of the moisture and the water that's in there as well, creating different weather patterns. So if you're altering the pattern of the hot and cold air on the earth by increasing CO2 levels in the atmosphere, then that has the capacity of changing the weather patterns. And that's basically what El Nino is, and that's what global warming is about, is changing global weather patterns in ways that we can't predict and that we don't understand. And the idea is we want to understand that before, like, say, for instance, there's a massive weather shift and a famine starts to hit. That's what we don't want. That's what we're trying to avoid. So basically, kind of bring it all, technology is your application of scientific knowledge. Biodiversity is basically a word that we use. These are kind of buzzwords here. It's basically the total number of species, typically, that are alive. And of course, extinction is the death of a species, when a species completely disappears. And so that's kind of the loss there. So loss of biodiversity. In this case, we have ring-tailed lemur, who is endangered. They're native to Madagascar. You can actually see one of these guys at the Denver Zoo. So that's what we're talking about. So that's the end of chapter 1. And so what we will do then is I'll go ahead and post that, and then get ready for chapter 2.